Good morning. Do you hear
Good
me?
morning. Yes.
So the topic of the lecture is chemotherapeutic drugs and antibiotics. It is a special topic for pharmaceutical faculty students. What does it mean, chemotherapy? Now we will talk about treatment of infectious diseases. Chemotherapy is a specific treatment for infectious diseases using chemicals with different antimicrobial spectrum. Because of infectious diseases are caused by different microbes, such as bacteria, viruses, fungi, and protozoa. So for treatment of infectious diseases, specific preparations are used. They should act on certain causative agent. And preparations, those are used in medicine, are differ by antimicrobial spectrum. Antibacterial preparations and spectrum is different. Some of them act on gram-positive bacteria, some of them act on gram-negative bacteria. Special preparations, medicines are antiviral. And special preparations are antifungi for treatment of disease caused by fungi mycosis. And specific chemicals are used for treatment of protozoal infections, antiprotozoal. All of them, they are different, and they differ by spectrum of action. Spectrum of action of medicine may be narrow and may be broad. Narrow spectrum drugs are active against small groups of microbes. For example, they are penicillin, nystatin. Broad spectrum drugs are active against large groups of microbes. For example, antibiotics such as streptomycin and tetracycline. Antimicrobial drugs against microbial pathogens differ by effect. Some of them have microbicidal effect and another have microbistatic effect. What is the difference? It's important, and the effect is described in every instruction for every medicine. It is written what effect has got this preparation on microbes. So what does it mean? Microbicidal. Microbicidal action leads to the death of microbes. So finally they will be eliminated from human body. Microbistatic inhibits growth and reproduction. It means in the presence of this preparation, microbes are not able, microbes those are sensitive to this preparation, they are not able to grow and they're not able to reproduce, but they survive, they're still alive. And when the drug is removed, when the doctor will cancel preparation and drug is removed from body by urine, microbes restore viability. They will multiply again. So it is the difference in action. Criteria for the effectiveness of medicines. Chemicals did create many toxic substances. Those are toxic for microbes. And by using of some of them, it is possible to kill immediately all microbes. But on the one hand, the chemical should be toxic for microbes, but on another hand, we are not going to use some harmful effect on human body. So it should be safety for the patient. And chemicals produce a lot of toxic substances, approximately 1,000, maybe more. And from that toxic substance, just a few finally are used for treatment of infectious diseases because of they should be checked by the criteria for the effectiveness of medicines. And chemotherapeutic index is used and it is the ratio minimum therapeutic dose to the maximum tolerated, and this ratio must be less than 1. So minimum therapeutic dose should be a minimum of minimum, and maximum tolerated dose should be more and more. In this case, and it should be less than 1, and reverse ratio means maximum tolerated dose to the minimum therapeutic dose must be more than 3. So maximum tolerated dose should be 3 times more and more than minimum therapeutic dose. Anyway, all medicines, those are used for treatment, not just treatment of infectious disease. They have toxic effects on human's body, of course. Complications are described in instruction for every medicines. Before using of some medicine, the patient should think about complications. So the next criteria is achievable serum concentration. Depends on the body weight of the patient, the dose of drug, ways and schemes of administration, as well as the rate of its excretion from the body. It's very important to think about this available serum concentration because of many medicines the patient took per os and preparations go through digestive tract, and they go through digestive tract, and just a small amount, a small concentration of this medicine is circulated in blood. They should go to the blood, but just a less amount of them is circulated in blood, and finally, by the way through digestive tract, they kill all normal microbial flora that exist in intestine, and they go out. So the concentration in the serum is maybe sometimes not therapeutic. Of course, it is better to use medicines for IV injections. In this case, or maybe intramuscular injections anyway, because of in this case, the concentration of medicines in the serum of blood is more. And could you imagine if the patient has otitis, for example, the inflammation in the ear, and the patient just took preparations per os, preparation goes through digestive tract, and it is far, very large distance from intestine to the ear, and of course there is no the proper concentration of the medicine in the serum of blood. So now preparations are produced to create a high concentration in the blood, but not all of them are able. And not all of them are able to go inside the human being cells to kill bacteria. Those are able to survive inside the human being cells. I'm talking about obligate intracellular parasites like rickets and chlamydia. They constantly multiply just inside the human body, so they're not available for all antibiotics and chemicals. Not all of them are able to go inside the human being cell, and some species of bacteria are facultative intracellular parasites like Brucella, like gonococci, meningococci, tuberculobacteria. So most of them are not available for medicines. And we should think about it. The third criteria is a spectrum of activity of antimicrobial agents. So antimicrobial agents act only on vegetative cells and not on spores of bacteria or cysts of protozoa. So we should realize, and the antimicrobial agent must penetrate the microbial cell, contact the corresponding target, and modify it. At the same time, retain its structure or form and an active metabolite. So for every antibiotic or chemical, there is special target for action. Target for action is some reaction inside the living cell. Cells among microbes are bacteria, they have cell structure, fungi have cell structure, and protozoa. So it is possible to develop some antibiotics or chemicals to destroy some reaction, As for the viruses, I hope you remember that viruses have no cell structure. And if the virus particle is out of the human being cell, of the host cell, any host cell, there are no any reactions in this virus particle, no any metabolism, so no any target for action. So it's very difficult to create medicines for treatment of viral infections. The viral has some properties of like living particle if a reproduction of viruses goes on inside the host cell. But in this case, virus did use ribosomes of host cell, and in patient body, reproduction of viruses is going on inside the human cells, and the human cell produce enzymes. Those are necessarily for viral reproduction in accordance with viral genome. And if we will destroy viral reproduction, anyway, finally we will interrupt the binary division of the human being cells. So it's dangerous. So because of this problem, we have just a few chemicals, not too many, for treatment of viral infections. And they are special again. They are specific for every viral infection. Okay, so chemotherapeutic agents in general are divided into two groups: antibiotics and synthetic antimicrobial chemotherapeutic drugs. They are not the same. Then I will explain you what is the difference. Synthetic antimicrobial chemotherapeutic drugs are they are artificial substances. They are produced by chemists, and they belong to different groups: sulfonamides, nitrofuran derivates of hydroxyquinolones, quinolones, fluoroquinolones, nitroimidazoles, allylamines, and oxazolidones. It is very important for pharmaceutical faculty students to know these preparations. Antibiotics. What are they antibiotics? Antibiotics are medicinal products of biological origin, selectively inhibit reproduction or kill microorganisms. Antibiotics are produced by living organisms. They have natural origin. All antibiotics have natural origin. They are produced by different living organisms, such as microbes, plants, animals, and humans. Antibiotic classification. Antibiotics, number one, antibiotics are classified by origin. Antibiotics are received. Do you remember the first antibiotic, penicillin? Penicillin in Latin, penicillin in English. Anyway, you will learn by heart finally Latin names if you are going to be a pharmacist, so you should know Latin names. So, and the first antibiotic was produced in USA at 1940. It was named penicillin, and it was produced from fungi, Penicillium genus. Molds, those belong to Penicillium genus. They did produce commercial industrial penicillin. It was the first antibiotic. And nowadays it's still produced by these fungi. And bacteria that belong to Actinomycetes genus, they produce a lot of antibiotics. They are very active producers, and so they are written separately. For example, streptomycin, kanamycin, gentamicin, produced by Actinomycetes. And other bacteria are able to produce some antibiotics like polymyxin, for example. And some antibiotics are produced in animal's body, animal origin, like lysosome is produced in animal's body, and lysosome is produced in human's body too. In saliva, lysosome is produced, and lysosome is a industrial preparation. A lysosome is produced from the hens and egg because of in the hens and egg there is a substance lysosome, and such medicine is produced now. It is possible to buy in any pharmacy. And plants are able to produce some substances. Those are able to inhibit the multiplication of bacteria. And for example, chlorophyllite, it is produced from eucalypt trees leaves, and it is very effective preparation against staphylococci. It is produced in the oil for mucosa membrane like nose or throat, and it is produced in ethanol like tincture ethanol for the skin. Very effective against staphylococci. So classification by origin, then the second antibiotics are classified according to the method of obtaining. Some antibiotics are natural. They are named biosynthetic preparations. Benzyl penicillin, for example, and chloramphenicol. They are produced and they are used like they are produced by producers. But nowadays, unfortunately, due to using of antibiotics, microbes did develop resistance, acquired resistance, and now it is necessary to change the chemical structure of antibiotics, and such antibiotics are named semi-synthetic. An example is ampicillin. The structure is of natural antibiotic of penicillin is changed a little bit. And nowadays, some antibiotics are produced by chemists. They are named synthetic antibiotics. Because of the chemical structure of natural antibiotics is known, and of course it is difficult to cultivate producers and then to isolate to get purified antibiotic. It is better to produce antibiotic by chemical way now. Like chloramphenicol is natural antibiotic, but synthetic antibiotics is analog of natural antibiotic against obtained by chemical synthesis. So, according to the mechanism of action, antibiotics suppress various processes in the living cell. There are targets for action of antibiotics. Number one, some antibiotics are able to inhibit the synthesis of cell wall components, like penicillins and cephalosporins are able to inhibit the production of peptidoglycan. And gram-positive bacteria have a lot of peptidoglycan in the cell wall, and in the presence of such antibiotics, they are not able to produce peptidoglycan and they become protoplast. Bacterial cells are those lost the cell wall completely. And if gram-negative bacteria are in presence of such antibiotics, they are not develop peptidoglycan, but they are able to develop lipopolysaccharide layer protein, liposaccharide layer that is above peptidoglycan. So they don't lost completely the cell wall, they lost partially that peptidoglycan layer, and they are named spheroplast. Do you remember? Some of them are not able to survive, but some of them are able to survive and multiply, and so, they are named LFOPs. Such bacteria, those are able to exist without the cell wall, and they have cell wall by their nature. And L forms are not stable. Some of them are non-stable. And if doctor will consult penicillin, these L forms are again will produce peptidoglycan, so they are able to reverse to original shape. They are not able to produce peptidoglycan just in presence of penicillins and cephalosporins. Another bacteria are able to act on to destroy functions of cytoplasmic membrane, such as nystatin and amphotericin B. They are antifungal preparations, and the target of action for them exactly are sterols. Those are in cytoplasmic membrane of fungi. Fungi are eukaryotic cells, so they have sterols, ergosterols, in the cytoplasmic membrane, and due to action of these antibiotics, cytoplasmic membrane is destroyed and lysis is developed. Unfortunately, human being cells are eukaryotic cells too, and they have the same sterols. So antifungal preparations are very toxic for human's body. They are harmful for human being cells at the same time. Some bacteria are able to inhibit the protein synthesis on ribosomes, such as streptomycin, tetracyclines, and erythromycin. And number 4, some antibiotics are able to inhibit the replication of nucleic acid synthesis, such as rifampicin antibiotic. Then antibiotics are classified by chemical structure. By chemical structure, they belong to different groups of antibiotics, and there are group names and there are examples. And you should memorize because of then for doctors, sometimes instead of names of antibiotics, just names of groups are written. So you have to know what antibiotic belong to every group. Exactly for you, pharmaceutical faculty students, you should know. So group is named beta-lactams, because of they have beta-lactam ring in the chemical structure. Like penicillins and cephalosporins are named beta-lactams. Then macrolides and azalides includes erythromycin, oleandamycin. Group named aminoglycosides includes streptomycin, gentamicin, kanamycin, tobramycin. Then the group is named tetracyclines. Tetracyclines and includes oxytetracycline antibiotic, doxycycline antibiotic. The next group is named polyions. Polyions includes antifungal preparations such as nystatin and amphotericin B. Ansamycins like rifampicin and rifamin, and additional class chloramphenicol and griseofulvin. Chloramphenicol and levomycetin is another name. Now, what about synthetic antimicrobial chemotherapy drugs? Synthetic. Again, they are divided by chemical structure on different groups. For example, sulfanilamide preparations includes sulfadimidin, sulfatiazole, and sulfadimetoxin, and so on. They have bacteriostatic action. Bacteriostatic action means, I did explain you, bacteria are not able to multiply in the presence of such preparations, but they don't die. Mechanism of action: anti-metabolites inhibit the synthesis of purine and pyrimidine bases. The spectrum of action is wide: gram-positive and gram-negative bacteria, fungi, and protozoa. The next group preparations, derivatives of oxyquinolones, includes nitroxaline, then quinolphone, etatrix. They have a bactericidal effect. Bactericidal effect means bacteria are not able to survive, they will die in presence of these chemicals. Mechanism of action lead to disruption of energy processes in the cell. Spectrum of action: gram-positive and gram-negative bacteria, some protozoa, and fungi from Candida genus. They are used for treatment of candidiasis also. The next group, nitroimidazole derivatives, includes metronidazole, tinidazole. They have a microbicidal effect. Microbicidal effect means they are able to kill microbes. Mechanism of action: disrupt the structure of DNA. The spectrum of action is wide, includes protozoa, gram-negative anaerobic bacteria, and by the way, metronidazole acts on gram-positive anaerobic bacteria too, unfortunately. Then I will explain you why it's not good. Allylamines include terbinafine, trade name is another lamizil, and they have microbicidal action, means kill microbes. Mechanism of action: destroy of the cytoplasmic membrane of the cell. Action spectrum: fungi. Fungi. They are used for treatment of disease caused by fungi, very often for treatment of dermatomycosis. And then there are special groups of medicines. Those are used for treatment of 13 infectious diseases, like antisyphilitic drugs, for example, for treatment of syphilis. Benzyl penicillins are used. Fortunately, treponema, causative agent of syphilis, treponema pallidum, did not develop resistance to benzyl penicillin. Now it is used almost 60 years, and we are lucky in this case. Treponema did not develop resistance, so for treatment of syphilis, benzyl penicillin is used. Penicillins, they are able to accumulate it in human body, and if patient has allergic reaction to these preparations, another antibiotic should be used, like tetracyclines, for example, and bismuth preparations, biquinol and bismuthirol, for example. Then the special group of medicines are anti-tuberculosis drugs used for treatment of tuberculosis. It is, of course, multi-drug therapy for treatment of tuberculosis. The patient will get a few preparations, including antibiotics and chemicals, and unfortunately, tuberculosis bacteria did develop resistance to many medicines. Those are used for treatment. One day, the strain of tuberculosis bacteria was isolated from the patient with tuberculosis in USA that had resistance to 32 medicines. Those are used for treatment of tuberculosis. How it is possible to treat such patients, okay? Unfortunately, strains are able to become resistant. There are just a few medicines. Those are used for treatment of tuberculosis, such as rifampicin, antibiotic, streptomycin, antibiotics, aminoglycoside, kanamycin, belong to aminoglycoside group. And there are some chemicals, isoniazid, ethambutol, and other. So just for example. Now I will make a break, and then in 5 minutes we will continue. Are you able to the same link? It is constant link. So in 5 minutes, by using of the same link, please enter the lecture again. Do you hear me?
Yes, okay.
Okay, so
Yes, yes.
5 minutes break and then we will continue.